All right. So, hello everyone, and a warm welcome to all of you. Thanks for joining in today. I am Ritama. I'm currently a postdoc at University of Pennsylvania, and I was an ex-BSMS student of ISER Kolkata. So it's truly an honor to be back, and I'm truly delighted to moderate today's session, which is about the Nobel Prize in Medicine 2020, which was awarded on the discovery of hepatitis C virus to Dr. Charles River, Dr. Michael Howton, and Dr. Harvey Alter. Prior to their work, the discovery of hepatitis A and B were already a step forward. However, the blood transfusion, post blood transfusion cases of hepatitis were still unresolved. So that led the way to the discovery of a third hepatitis virus, which is the hepatitis C virus. Uh, just like the other hepatitis virus, this strain of hepatitis C virus also primarily affects the liver, though the chronic and the initial infection phase doesn't really have any symptoms, but a long-term effect may lead to may result to liver failure and also sometimes liver cancer. So it's quite a deadly strain of amongst all the hepatitis virus, and vaccines are still on the way. However, this discovery was very significant in limiting the blood transmitted hepatitis cases. So, and after this discovery, almost the cases for post blood transfusion related hepatitis cases reduced from 30% to almost zero in the 2000s. So this was a phenomenal discovery, and we'll learn more about this from our guest lecturer today, who is Dr. Soumitra Das, and we'll begin with his talk now first.
Thanks for the invitation. So it's really a pleasure to join this science communication series of ISER Kolkata. I'll be happy to present on our the journey with the hepatitis C virus. Particularly this year is very relevant because the Nobel Prize for the Physiology and Medicine has been awarded to the group of scientists who actually are the champion and the pioneer behind this whole hepatitis C virus discovery and its impact on the global health and the development of the therapeutic approaches. So before I start, I'm currently at the National Institute of Biomedical Genomics. We joined here in 2018. Before that, I've been working on hepatitis C virus for last 22 years in the Indian Institute of Science, Bangalore. My lab is still continuing there, and they are actually contributing to the field of hepatitis. Now, just to start with, as we wanted to have it very basic thing, I was just looking at the historical perspective. I mean, what we know about hepatitis. Now, if you really look at the literature, this is almost like a 3000 BC. Sumerians they used to think that the liver is the home of the soul, and the jaundice is the attack on the liver by a devil named Azau. Then 400 BC, Hippocrates actually said that the bile contained in the liver is full of flame and blood and erupts. After such an eruption, the patient soon raves, becomes angry, and talks nonsense. Then there is a mention in 17th to 19th century where the jaundice epidemic was actually noticed in the military, in the main and the civilians. And in the French camp, because of the yellowing of the camps or the battlefields, they used to call it as a jaundice champ or the battlefield jaundice. And the clinical term was icterus. Icterus is the yellowish or the discoloration of the skin due to hyperbilirubinemia. And actually, funny thing is, at that time, the myth was that the icterus is actually Icterus galbula, is actually a Baltimore oriole, which is a yellow bird. And the myth was at that time that if you develop this kind of a yellowish in the skin, you go to the forest, sighting the bird was supposed to cure a person with jaundice. So while these things were going on 17th to 19th century, there was an accidental discovery of the Australian antigen by Dr. Bombard. And he ended up discovering actually he was trying to study the genetics of the disease susceptibility and ended up with a strange antigen, which they named it as an Australian antigen. And that was the discovery of the first hepatitis antigen. Now, hepatitis, as you know, can be actually triggered by different things. Like it could be because of the alcohol induced, it could be an autoimmune disease, disorders, non-alcoholic steatohepatitis. Many a times it is due to the toxins or other infections. But I mean, what we were actually more concerned now is the viral hepatitis, and which is actually coined by several alphabets, A, B, C, D, E, F. There is no F, but there is a G. So the F, hepatitis F, could not sustain. I said hepatitis F failed. That's how now we jump to hepatitis G virus. So as you see here, all these alphabets, A, B, C, D, E, and G, they are actually all causes viral hepatitis, except hepatitis B and hepatitis D. They are RNA viruses. Hepatitis B is partially double-stranded DNA, and hepatitis D is also a single-stranded RNA viruses. But it needs a satellite viruses like hepatitis B to carry on the infection. Now, as you see here, hepatitis B was a major problem, 240 million chronic infections globally, and hepatitis C is also following the hepatitis B, 150 million chronic infections globally was actually being noticed. Now, as early as in 1989, hepatitis C is known to, it was known to cause chronic hepatitis, liver cirrhosis that leads to hepatocellular carcinoma. And there is some estimate in 2007 which shows that it actually suppresses the HIV infection to some extent. And HIV was actually 12,734, where hepatitis C was 15,106. So it's a major concern. Now, most of the time, the hepatitis C virus infection causes the chronic hepatitis. But as you see here, 15 to 25% people are lucky and they can actually clear the virus. But the 75 to 85% which actually develops the chronic hepatitis can lead to, of that, 20 to 30% into a cirrhotic liver, and 2 to 7% of that can actually end up with the hepatocellular carcinoma, which is actually an end-stage liver disease. How people get hepatitis is actually most of the time it was because of the injecting of illicit drugs, high-risk behavior in the past, sexual activities, and sometimes there were actually blood transfusion, particularly because of the hemodialysis and the healthcare employment, people also used to get it. And before 2000, a lot of people got it in India because it was not mandatory for the blood donors to check for hepatitis C. So it was actually in our population. And as you see, hepatitis C virus, this is a 2013 picture. It's almost like an all over the world. And in some of the pockets, there are actually huge incidents of hepatitis. And there are several genotypes. There are 7 genotypes so far, and there are 62 subtypes, and it's still emerging and it keep on mutating. It's a major problem. Now, the thing that we are going to discuss it is these 3 persons, the Harvey Alter, Michael Howton, and Charles Rice. These 3 people were actually saved the whole world from this menace of hepatitis C virus. How their discovery led to the treatment is actually I'm going to highlight in my talk. So let's start with the Harvey Alter's contribution. So as I told you that hepatitis B was actually discovered, Bloomberg was actually given Nobel Prize in 76, I think, if I'm not wrong. And then, but there was still the problem persisted. Means there were actually incidents of blood transfusion after even screening of hepatitis B. So people were actually termed as this is a non-A, non-B viral hepatitis or NANBH. Now, this virus is actually was causing a major concern. And Harvey Alter's group, actually, it was a landmark paper in the Lancet in 78, where they have shown that if you actually take this serum from an infected person, which is actually non-A, non-B hepatitis, and inject it into chimpanzee, you can see a similar kind of a hepatitis developed in chimpanzee. So as you see here, uninfected and infected chimpanzee, chronic infection was more frequent compared to the hepatitis B infection. And it was, as I told you, it was named as a non-A, non-B viral hepatitis. Serum from patients with acute or chronic non-A, non-B hepatitis could transmit the disease to chimpanzee. And they also actually pointed out the virus is somehow coated with the liver droplets. The virus contains lipid, and the size was actually ranging around 30 to 60 nanometer. This was actually the first report on the presence of this non-A, non-B hepatitis. And then Michael Howton actually continued research and tried to identify what is this non-A, non-B hepatitis causing agent. So what they did is essentially they collected the plasma from the infected chimpanzee, spun it in the ultracentrifuge, deleted it, and then isolated the DNA and RNA from there. And that DNA and RNA, they did actually a shotgun and then expressed in a lambda GT11 cDNA library. They made a cDNA library of lambda GT11. And then they actually probed it with the antibodies that were isolated from this non-A, non-B hepatitis patient. And then they actually screened for the clones which are actually expressing the proteins that can be neutralized by the antibody or lighted up with the antibody that is collected from the patient. And they were successfully able to determine the properties of a clone. They named it as 511. This they found that this is an extra chromosomal derived from the RNA found only in the non-A, non-B hepatitis samples and distantly related to flavivirus, encodes a protein that binds to the antibodies found only in the non-A, non-B infections. And as you see here in the Western, they actually clearly showed that it's actually specific for the antibody isolated from the non-A, non-B hepatitis, but it's not lighting up with the hepatitis B or hepatitis A. So this is essentially, again, another landmark study by Chu et al. From the group of Michael Howton, which came out in Science in 1989. Now that's 1989, and then Charlie Rice took it over, and then he tried to identify, is it possible that you can characterize this hepatitis C virus? So that's the time they started calling it as a C, because we already have hepatitis B, we have hepatitis A, now they named it as hepatitis C. Now, the question is whether the hepatitis C alone can cause the hepatitis or not. So Charlie's group showed, which was actually published in Science in 1997, that if you can isolate and clone the virus from an infected patient, and they showed that if you have this RNA and tagged it with a conserved 3' end, non-coding regions at the 3' end, that engineered RNA, when they are actually try to incorporate some mutations which will inactivate it, or you use the wild type and try to do this experiment in chimpanzee, and they could show that actually they are able to cause the infection. So that was the first incidence of a clone of hepatitis C genome, and people, their group has demonstrated that you can make the infectious RNA from the clone, and that infectious RNA itself can cause the disease. So if you can inject this to chimpanzee, it can express the viral protein, replicates, and then he has actually recapitulated this whole thing in a cell culture model. This is very important. As you know, that after the discovery of the virus, it's very important whether you can have a cell culture so that you can continue to understand the biology of the virus. So actually, Charlie Rice's results opened up a huge window to work on this virus, to look at the biology of the virus, to understand the mechanism of pathogenesis of the virus, which was not possible earlier. And this was done in 1997. So essentially, how this discovery, you can see the roadmap. The first, there was actually a report on an observation of a non-A, non-B hepatitis virus in the blood transfusion patients. So when this was actually taken it up and then infected in chimpanzee, it was found to come up with the similar kind of a disease. Then this was actually, this plasma was then probed for the DNA and RNA, and that genetic fragments was actually cloned, and then it was actually probed by the antibodies infected isolated from an infected patient. And finally, the genome was identified, and Charlie Rice showed that you can actually have a cDNA by reverse genetics, and that infectious cDNA, you can make the RNA in vitro, and that RNA can actually causes the same hepatitis C disease in a cell culture and a chimpanzee model. So this was the discovery, and what this discovery led is essentially now you have a handle to detect the virus. Remember, this was 1997, and from 2000, it was so popular for detecting the hepatitis C virus, because previously it was not very popular test to find out hepatitis C in the, as I told you, in the blood donors or in any other blood transfusion cases. So it became a normal practice for the people to test for hepatitis C virus. And that actually led the research to the eliminations of the risk of transfusion-associated hepatitis, and that led to the research to identify and discover antiviral drugs. As you know, now there are several anti-hepatitis C drugs are available over the counter to actually combat this particular disease. So this disease can now be cured and raises potential for the global eradication of hepatitis C virus. Actually, incidentally, in the last meeting when we attended hepatitis C, we used to have a big meeting, and Charlie Rice actually declared it a couple of years back that we hope that after 2020, we don't need to have a hepatitis C virus meeting anymore, because, you know, majority of these things are now under control. While saying so, we also appreciate that there are a lot of things are still not known, and I'm going to highlight that part. Now, I'll try to give you a little glimpse of research that our lab have been doing it. This was discovered in 1989, and I think joined a lab in UCLA in 1991. And from then, I have been working on hepatitis C virus. So from the beginning, my particular journey of research started almost at the same time when the hepatitis C virus research was evolving. So I'll just give you some glimpse of results that we have actually generated in the laboratory at Indian Institute of Science. Now, hepatitis C virus, as I told you, now we know a lot about it. It's an RNA virus. It actually goes inside the cells and then uncoat. The viral RNA makes viral proteins, and that's actually called translation of the viral protein. And then these viral proteins binds to the viral 5' UTR and the 3' UTR, and then it's in a membranous web structure. They make a replication. First, from the positive strand, there is made a negative strand, and that negative strand is actually used more and more as template for more and more positive strand, which is ultimately packaged, we call virion assembly, and then the virus is released out of the system, and then it actually infects the other neighboring cells. Now, what is interesting about is that hepatitis C, or any for that matter, are actually restricted to liver. That's why we call hepatitis. Hepatic cell, they actually target the hepatic cells. And for hepatitis C, it is even more specific that it infects only the hepatocytes, but it doesn't really affect that much of the stellate cells or the Kupffer cells, which are neighboring cells. So it is something you can imagine that very specific for the entry. That means somebody is trying to get inside the room, and once they get into the room, they know exactly where to go. They ended up into a kitchen, which you can take it as a hepatocytes within the big house of liver. And why in the kitchen? Because you have the utensils that are required to make good food, so that the virus can actually survive inside this liver. And that food is really palatable. And as you see, that this sub sandwich are actually cut down by knife, and there are cellular protease, and the viral protease actually chops this viral polyprotein into structural and non-structural protein. There are several structural protein like the core, envelope, P7 is an ion channel protein, and the NS. And there are non-structural protein like the NS2, NS3, NS4A, NS4B, NS5A, and 5B. NS5B is the RNA-dependent RNA polymerase, and NS3 protease is the protease which actually cleaves the viral polyprotein into small pieces of structural and non-structural protein. Now, arguably, people will always try to inhibit the viral protease or the RNA-dependent RNA polymerase so that they can inhibit the viral replication, and that's what it is done. As you know, initially hepatitis C virus was actually treated with pegylated interferon and ribavirin, which is a nucleoside analog. But later on, as I told you, with the discovery of the cell culture and the advent of the progress of the research, people started screening for different compounds. And now there are telaprevir, and which actually inhibits NS3 protease, and sofosbuvir, which actually inhibits NS5B polymerase, and the NS5A is actually targeted by ledipasvir. So these are the compounds which are now turned into a drug, and that can actually eliminate or bring down drastically the virus titer in an infected patient. So that is the good sign. But as for everything, there is always limitations in the drugs, because as I told you that hepatitis C has 7 genotypes and several subtypes. So the genotype specificity is an issue. Emergence of drug resistance is another issue of the mutants. Relapse of once the drug is stopped. There are several cases now coming up that even after the virus titer is down, after 6 to 12 months, the people is coming back with the hepatocellular carcinoma, or actually there is a relapse of the disease. So additional therapy associated side effects are also there. So there is a scope to actually contribute to research to find out alternative drugs. Now, I will just give you just a short glimpse of what we are doing in our laboratory at the Indian Institute of Science, Bangalore. Now, when we actually started my laboratory in 1998 in Indian Institute of Science, at the same time, there was a paper in Science which showed that actually in the ribosome, that usually the cellular RNA binds to the P sites and the A sites, but hepatitis C, interestingly, binds to the exit gate of the ribosome. So if you really look at, this is the head, this is the platform, this is the wick, this is the exit gate, where there is a pink place, there is a protein called ribosomal protein S5. We showed that actually this protein interacts with the viral RNA, and the virus gets entry into the ribosome, triggers a conformational alteration, and access to the A sites and P sites to generate their protein synthesis. So it is definitely an unique mechanism of viral RNA translation. Although it's an eukaryotic RNA, you can still see there is a differences with the eukaryotic RNA translation and this viral RNA translation. So, and as we mentioned, that there is a ribosomal protein S5 which is involved for the viral RNA interaction. But this procedure, it's unique and fundamentally different, and it involves a lot of host factor, not only the viral protein. There are some viral proteins involved, but there are some host proteins involved. So my lab was actually interested to find out which all ribosomal proteins involved for the interaction with the viral RNA, for the entry, which all host factors are involved in the translation and replication of the virus, and which all viral proteins are actually necessary to regulate the translation and the replication of the virus. Ultimately, is it possible, knowing this biology and the mechanism, can we target to design a selective antiviral? Remember, as I told you, that the translation is unique and fundamentally different. So if you can really target it, we don't expect any side effects. Like an NS3 protease and NS5B, when you actually inhibit, you ended up actually inhibiting a lot of other cellular protease and polymerases. So this kind of effect we can actually avoid if we have a very specific targets. But while understanding this biology over the years, we have been actually studying that several host factors. And interestingly, some of these host factors are nuclear protein. They have nuclear function, but in the infected cells, they come out from the nucleus to the cytoplasm, bind to the 5' UTR. And in the 5' UTR also, the translation is something different. The ribosome binds to an internal sequence closer to the initiator AUG, which we call internal ribosome entry site. So unlike cellular RNA, where there is a cap structure at the 5' end, ribosome binds to the cap structure. In this case, hepatitis C, ribosome 40S subunit can directly go and bind to the viral RNA near the initiator AUG. And this is actually facilitated by several transacting factors or host factors like La and PTB. We showed that some of this protein also binds to the 3' UTR and causes a switch from the translation to replication. And not only that, there are now microRNA like MIR122. We also showed MIR125B. They are regulating the abundances of their targets, and those proteins, RNA binding proteins, and their affinity towards binding to the 5' UTR and 3' UTR. Now it is actually looks like a big concert where there are several players, and latest players along with the HUR, PTB, and La protein that we have been working, we found there are microRNAs and there are long non-coding RNAs which are actually contributing to the HCV orchestra in the infected cells. And over the years, we have shown how HCV infection, there are several proteins and the host factors, viral proteins and host factors are involved. The microRNA and the LNC RNA that are involved, as I told you, La, PTB, HUR, or the MIR122, 125B. And very recently, we showed the Hulk, which is a liver-specific long non-coding RNA and highly upregulated in the liver cancer. And actually, we showed that how it actually helps in the virus to increase the lipid droplets and package it along with it and use the VLDL pathway to excrete out and then affect the neighboring cells. But now what is unsolved mystery is the hepatocellular carcinoma, and we are actually trying to understand how P53 and other players are actually contributing to the hepatitis C virus induced hepatocellular carcinoma. So we started with a simple observation with a translation that we call protein synthesis and tried to make it translational. How did we do it? We showed there is a small RNA which can prevent the ribosomal RNA, hepatitis C RNA and the ribosome interaction. We showed if you just take a peptide corresponding to the RNA binding motif of the La protein And this 7-mer peptide, which actually forms a beta hairpin structure, is good enough. If you just tag it with the 6 arginine residues, it goes inside the cells and can stop the virus RNA replication. We also showed that NS3 protease can also bind to the viral RNA for the switch from translation to replication. If you use a peptide corresponding to the RNA binding motif, it can prevent the viral RNA translation. Along with that, because of the suggestions from the government of India, we were also trying to see some compounds from pomegranate and also Phyllanthus summer as natural sources. And we have identified the pure compound from the natural sources. They can inhibit the hepatitis C virus replication to the extent that we are actually getting from the commercial drugs. And we also found some monoclonal antibodies. This in collaborations with Professor Anjali Karande's lab and Dr. Soma Das, and we showed that these monoclonal antibodies can efficiently prevent the virus entry. And very recently, we have made a virus-like particle with the hepatitis C virus structural protein and showed that how that VLP can be used as a vaccine candidate. Now, all these things we are testing it. As I told you, that hepatitis C virus doesn't have any animal model. It infects chimpanzee, but that chimpanzee work is now discouraged, and it doesn't infect mouse. But we have collaborations with Japan with some laboratory where we have mouse with a harboring human hepatocytes, and this mouse can be infected with the viruses. In this system, actually, we tested all our antiviral compounds and see whether we can get reasonable results in an in vivo model. So what still remains is an appropriate model. As I told you, chimpanzee work is now discouraged. Mouse model, we are using humanized mice, as I told you, and some researchers are trying ectopic liver generation to understand the virus biology in an in vivo system. Vaccines made different types, but there are limitations because of this viral sequences, genetic variations, because of the error-prone replication by the that RNA-dependent RNA polymerase, which is called the NS5B. Extensive glycosylations of the viral envelope protein also keeps changing its immunogenicity. And the major hurdle is the limited production of the whole inactivated virus. We still can't make tons of viruses as we do for other viruses. For some reason, there is a roadblock in that area. And the major thing that is actually being addressed is the progression to the hepatocellular carcinoma. You know, many a times after the viral infection, several pathways are activated. Unless those pathways are restricted, just bringing down the virus titer does not really prevent the disease progression. So we are actually trying to find out how we can prevent the disease progression. The patient who are curing of the virus should not develop hepatocellular carcinoma. That's a big challenge ahead of time. And the epigenetic modulations of the proteins and the nucleic acids, and trying to see how they can actually contribute to the hepatocellular progression. It's possible that during viral infection, these epigenetic changes happens. Unless we identify that which epigenetic changes are actually contributing to the disease progression, it is difficult to really take care of this hepatocellular carcinoma disease, and triggered the secretory molecules such as that exosome, its combination and composition. Finally, the last words that I got it from a message, none other than Charlie Rice. He is also saying that let's hope that this, that their effort, and he is appreciating that all over the world there are several HCV researchers are actually contributed to this final outcome that we could actually handle this disease efficiently. But let's hope that this example of the benefit that science can bring will encourage the public to embrace and support fundamental and curiosity-driven research. This is the key word. The curiosity-driven research is something which all of us are actually encouraging. And as we are in the midst of the experiencing with the ongoing pandemic, there are many challenges ahead. And this is one such example. We should not be scared away from the menace of RNA viruses. This is one example that it was discovered in 89, and now in 2020, actually, we could actually think of eradication of the disease. So I'm sure many of these RNA viruses we can actually handle, taking this as an example. So with this, I would like to thank the people who are working. These are my present group. These people are actually at NIBMG. They recently started working here at NIBMG, and these are the people who are working in my laboratory at the Indian Institute of Science. Our research is heavily funded by the Department of Biotechnology, DBT. We have a Center of Excellence for Hepatitis C Virus Research, which went on for 10 years. And of course, the support from the other funding agencies. Thank you all.
Thank you, Dr. Soumitra Das. That was such an enlightening talk. I would also like to thank Cogito 137 and the Science Club of Pfizer Kolkata for organizing this event. Moving on to the next segment, we have a small panel discussion where I will be joined by two more alumni, Dr. Shalini Singh and Shondipto, if I got that right. So I would like to request Shalini and Shondipto to give a small introduction about themselves.
Hi, this is Sandipto. Can you hear me? Fine.
Yeah.
Okay, so yeah, small correction. I'm not an alumni of ISAR, but I have really close friends, and I think Dhiman, a very close friend of mine, he kind of introduced me to the wonderful team that is doing a phenomenal job in arranging all this in time of in this unprecedented time, right? So having said that, I would like to briefly introduce myself. I started my research actually in University of Calcutta in Dr. Jogojyoti Chattopadhyay's lab, where I met Dhiman, and then there my research was on mostly RNA viruses, but then I moved on to University of Kansas Medical Center, where I was studying breast cancer progression in mouse models. And then my role was to basically look at a role of some extracellular matrix proteins in breast cancer progression. And now what I'm doing, now I'm in Cornell, in Weill Cornell Medicine in New York City, and trying to understand the role of gastritis, inflammation, and the role of microbiota in inducing gastritis, and how it eventually leads to gastric cancer. So that is my very brief introduction.
Thank you, Shondipto. Shalini, would you please introduce yourself? Shalini, are you there? We might have lost her. So Shalini was also was in the master's program of ISAR Kolkata. I kind of overlapped with her, and she worked with Rupak Datta for her master's thesis. And I think after that, she went on to IISc Bangalore to pursue her PhD. So now we'll have a small panel discussions where we'll take in some questions from the audience, and also we'll discuss. Shalini, are you there?
Hi, Ritama. Yeah, I got disconnected in between. So I just heard what you said. Yeah, I was there in Dr. Rupak Datta's lab with Ritama during my master's, and presently I'm a graduate student in Dr. Amit Singh's lab at Indian Institute of Sciences, Bangalore. So my work concerns trying to understand the pathology of HIV, and our group as a basic uh, it basically tries to understand how modulating antioxidant systems of host as well as pathogen alters the disease physiology in case of tuberculosis and HIV. So I'm working on antioxidant based nanoparticles which inhibit viral replication.
Okay, great. So let's get started with discussing some of the important science related to the HCV, hepatitis C virus. So let's first discuss the epidemiology of the hepatitis C viral infection. So basically, what are the geographical distribution of this virus? And are there any people at higher risk than others, people with some pre-existing conditions, etc.? So yeah, Shondipto, do you have anything to say on that?
Yeah. So about the geographical distribution that that you just so basically I was going through a few websites which are kind of world standards when it comes to uh, the statistics, the most recent statistics. We all look for, like, we all look up to WHO, and in whose website they basically say that it is the most affected regions are actually Eastern Mediterranean and European regions, which has a high prevalence rate of 2 to 1.5%. And then there are certain, and of course, as you mentioned, that there are certain populations or certain sections of people in the society or in the community who has a tendency, who are more susceptible to contract HCV infection. And since this is a blood-borne pathogen, we can all guess what are the different what strata of people, basically frequent drug users who uses syringes, and then people who had to go through blood transfusion, especially before 1990s, from what I understand, in India or all across the world. And also people, men who have sex with other men, which leads to a blood contact, in some cases are also at high risk of contracting this viral infection. So basically, like, in the end, people who are more prone to blood contact, since this is a blood-borne pathogen, are more susceptible in contracting this virus. I think Shalini can add on to this, because I guess it will have a huge overlap with the people who are susceptible to HIV also.
Absolutely, absolutely. So, as you said, any kind of people who are prone to very frequent blood transfusions, like hemophiliacs, people suffering from thalassemia, or undergoing hemodialysis, those are the more high risk populations, and drug users. To some extent, in some cases, mother to child transmission has also been observed, and HIV infection increases the risk of this vertical transmission. So HIV infection is one of the conditions which increases the susceptibility to this virus.
I see.
Yeah. Ritima, your mic is off. We cannot hear you.
Sorry, yeah. Audience can also ask questions. Please drop in your questions either in the chat section, or if you're comfortable in asking questions on the camera. Please go ahead. So I think we got one question. If questions are being taken, yes, kindly do throw some light on this. It's not really related to hepatitis C virus. I just wanted to know, why would retroviruses employ such an expensive replication machinery when other RNA viruses can go out about directly downstream producing mRNA and the protein and so on? Great question by Omkar Sahu. So, Shalini, do you want to take a try on that?
Well, to us, it may be that it's a more expensive machinery to replicate, but it's beneficial for the virus, right? Retroviruses are basically lentiviruses, which grow slow and persist for long. To us, it may seem that these are very tedious means of replication, but for HIV, it's perfect. We have known since, like, ever since the initial epidemic broke out, we have known this is the virus. Still, it's not curable. We cannot say that about RNA viruses. They are, comparatively, I would say they are easily targetable. Comparatively, it will differ from virus to virus, but it all depends. So these are evolutionarily selected mechanisms, and it works for HIV. The DNA is more persistent inside cells as compared to RNA. It's more stable.
Yeah, absolutely.
And if Shandipto has anything to add, you can.
No, you are the expert here in on
No, I don't
on
think.
on on retroviruses especially. So yes, I mean, like, we wish we knew why this virus behave like this, but yes, so, like, like, as you mentioned, what might look expensive to us, because we have the view, like, we have the view of how other viruses behave and can survive in a more economical way, if that is the right word. Then it might be kind of counterintuitive, right? But then I think, as you mentioned, this works for this virus. It has been selected by the evolutionary selection over years, and it's working fine, right? It's giving us a hard time, all of us.
Yeah.
So yeah, so why not?
All right. So, right at this time, we don't have any questions from the audience. So, I'll ask a question of mine. So, what is the key significance of this discovery of the hepatitis C virus? So, like the social benefits, I think Dr. Stromitra already touched upon that. But yeah, if someone missed that, we can discuss some of the key significances of the discovery.
Right. And sorry, so let me Shalini, do you want to start, or I can?
Yeah, anything. I feel the key significance here was that it made blood transfusion more safer.
Yeah.
Yeah,
Before,
true.
at this point of time, even after discovery of hepatitis A, which is non-blood mediated, even after discovery of hepatitis B, which is transmitted via blood, over 70 to 80% cases of this hepatitis, which was happening in blood transfused patients, those cases did not go down. So that was one major benefit because of discovery of this hepatitis C virus, HCV.
Yeah,
And again,
true.
Yeah.
yeah,
Yeah. And yeah. So so this discovery now have the stamp of the highest honor in science, right, on the land. And so, and then I knew about it, and I was just updating myself a bit more because of this also. And then all the Nobel Nobel worthy findings comes down to the single question of how many lives it saves, and how people are benefited, which basically means that how it makes us more safe. And as Shalini mentioned, I was reading somewhere that it was, and I think it was one of the scientists who mentioned that before discovery of hepatitis virus, I'm not sure whether it was before C or B, but in general, before discovery of the causing agent of hepatitis, it was almost like playing a Russian roulette. A significant fraction of the blood transfusion recipients were getting infected by this deadly disease. And more, the more scary part was more, and it is also the scary part now. In most cases, they didn't know about it for the longest time, and by the time they would be aware about it, uh, they are almost struggling, their liver is failing and something like that. So yeah, exactly. I mean, coming back to you, Ritoma, it is a important question that why is this discovery so important to us? Because it has saved millions of lives, and it is saving millions of lives even today. So
Yeah.
I was just looking at the like who statistics and who projects that we might be free of hepatitis in general, including hepatitis C, by the end of this decade, like 2030. So we are still 10 years back, like backlogged, right? So yeah, it, it is an extremely important discovery, just in terms of the sheer volume of the lives it has saved.
Yeah, true. And as I mentioned in my introduction, the number of cases post transfusion hepatitis cases, the risk before 1970 was almost like 30 to 40%, and that reduced to almost zero in the 2000s. So
Yes.
I would say it's pretty late to like award this Nobel Prize.
Yes, like
But
for hepatitis.
yeah, better late than never. So yeah.
Yeah, and
Okay.
the Nobel Committee was asked this question, that why did it take so long to to uh, to recognize the efforts, and then they just, I would say, they kind of hand waved, saying, oh, we take a lot of time in making sure that what has a role in humanity. And I think it was pretty obvious, quite like few years back, I mean quite a few years back, that it is going to be a game changer. So yeah.
Okay. So I'll ask another question since we still don't have any questions from the live audience. So this also, I think Dr. Das touched upon. So what are some of the basic preventive measures to stop the spread of the virus? So it's kind of intuitive. It's a bloodborne pathogen. So like having safe sex, and then trying to avoid illicit drug injections, and cleaner way to ethical and cleaner way to transfuse blood. These are some of the precautionary measures. If you have anything to add on that, and also on this same question, I would like to discuss a little bit on some of the methods to test the presence of this virus.
So, in case of preventive measures, as you said, what you said is correct. You require more of screening to make sure blood-borne transfusions are stopped, because that is completely preventable. That is one thing which you can reduce to 0% transmission through blood transfusion. And I think the need here is to uh, start run regular testing, especially in high risk people. I'm not sure how prevalent that is, because uh, I also I started reading little bit regarding hepatitis C virus recently, especially after the discovery and for this event, and I realized it's much more prevalent than HIV. But as you read and in general day-to-day activities, you don't read about HCV as much as you hear about HIV or tuberculosis. Almost 3% of population is infected with HCV. So still, and especially about India, many areas in the northern region belt, where in cases of villages, these diseases are prevalent. There are populations where almost 60 to 70% of population is infected, but due to poor communication, they do not even know that how it happened, or they do not know ways to stop it. So the need here is to start testing, make sure that you test high risk people. I think guidelines are there, but I don't know how frequently or at how diligently those are followed.
And do you want to throw some light on like what are the different detection methods to detect the
So,
presence of this virus in the blood?
so I think for most common method is by RT-PCR, RT-PCR testing,
Similar to COVID.
and yeah, and you can also test for antibodies. So seroconversion happens around 6 to 8 weeks after
Yeah.
infection exposure. So, and in most cases, the antibodies are maintained. In chronic patients, antibodies are always there because some amount of viral replication will always be there. And in case of asymptomatic patients who get cured naturally, in them also antibodies do persist for almost for years, like in some cases for lifelong. So,
Yeah.
True.
And thank God they persist, because
absolutely.
it was kind of uh, the antibodies that actually help to identify the virus. If you remember how they identified the virus, they basically threw on the serum of infected patients and were hoping that the antibody will identify the bacterial clones expressing the viral proteins. So yeah, thank God, thank God the antibodies persist. And yes, Shalini, exactly like any other virus, viral detection, and we that something that has come out of this COVID-19 situation is that people have become a bit more aware of what are the usual testing procedures, what's a virus, and many more. So and then my friends who are not even studying biology, or my parents or my family members, they know a lot, like way more about viruses, and then they know like most of the virus testings are done. It is either you have to identify directly the virus is present by probing for its nucleic acid, be it DNA or RNA, or you have to look for the viral antigen. So that is like a direct way, and an indirect way of knowing whether you had the viral infection sometime in your life, if the antibodies persist, is to look for the antibody titer. So yes, exactly what you said, Shalini. And but one quick follow-up question, Rituma, if I may, to Shalini.
Yeah, sure. Go.
Yes, yeah. So you mentioned that northern India has a like people in northern India are more exposed to this Hep C virus. So I was trying to look at what is the scenario in India, because how the map looks within India, like are there any high risk regions? So since you are a virologist in India, so I was curious whether you know of any red belts or red zone within India where people are suffering more.
So in India, actually, not very. There have been not very methodical studies regarding this HCV. There are few reports. There was one study in West Bengal, and then there have been few studies where they have checked in certain cohorts, and these are mostly hospital-based data. So mostly checked when blood transfusion is happening. So based on those reports, these
And yeah, you are
numbers
right,
are very.
and that was a very major critique of Indian biological science scenario during this COVID situation, was that we are not great in studying epidemiology of diseases, which should be very important in a country with such a highly like dense population. Yeah.
Okay. We have a very interesting question from the audience. Shushnato actually asked this question. So, given that there are so many different subtypes of this virus present currently, and even in the lack of a vaccine, detection and therapeutics has really helped in controlling the infections in case of hepatitis C. I would like to know about your opinion on the race to develop a COVID vaccine. So, Shalini, do you have anything to add on that?
Going by the standards, we are working. I mean, people are working really fast for COVID. A few years back, no one could imagine that within 1 year or few months span, you would start vaccine testing. So
I think what Shushnato is asking that for hepatitis C virus, even without the vaccine, just with like developing detection techniques and developing drugs, it has really helped to control the disease. But in case of COVID, we are running after looking for a vaccine. So yeah,
Because sorry, I mean, because no drugs have worked, right? We have threw in almost like what we had in our doctors. If you here in Will Cornell also, and also my friends in Delhi, AIMS, who were like at the very, who are basically trying to try everything, like everything that at their disposal, that has been shown to alleviate patients uh, with respiratory diseases. Those did not work, and then vaccine is the answer almost. I think we are also working, like scientists are also working towards a Hep C vaccine. So vaccine is the home run. I mean, vaccine is the ultimate success, right, against all of this. So why not?
prevention is better than cure. The old saying, I would say.
Right, exactly. And I would rather hope it would be the other way, that there will be a race for Hep C vaccine also. Now that it is in the limelight of Nobel Prize, that there should be race for all of these horrible infections which are killing people in mass, right?
yes. And I think simultaneously research is also going on
Yeah.
about like agents that can cure COVID infection. That's why every day Trump used to
Yes.
throw in all these drugs that
Yeah.
he
Yeah,
kind
Yeah.
of
it's on
heard.
beaten trial right now.
Yeah.
Yeah. Okay. So the next very interesting question that is coming to my mind is like, what are the key differences between hepatitis B and C viruses? And while one has a vaccine like readily available, hepatitis C vaccine is still not there. And why is it so? Like, what are the complications?
Shalini, you want to go in?
Uh, you go first.
Okay. So, so So I mean, I was reading about why it has been very like really difficult to develop a vaccine against hepatitis C, and then I was also looking at some basic biological differences between B and C, right? And then, as Dr. Dash mentioned, that hepatitis B is a DNA, it's a DNA virus. I think double strand, like partially double-stranded DNA virus, and C is a single-stranded RNA virus, and then uh, and then and then the common intuition is that kind of tells me is that which means that a Hep B virus would be less prone to variations, and quite rightly so, because if we look at the three top reasons that Dr. Dash mentioned, that why it has been a really difficult job to prepare vaccine against Hep C, where I was able to note them down, was first of all, like it was the high variability. It was like it is there are like 6 types and like 60 something subtypes. I think 7 types or 60 something subtypes. And then he also mentioned that there is extensive glycosylation on the viral envelope proteins, which kind of masks the epitopes. And also he mentioned that one of the major challenge, which I did not know and I also did not read about, is I think it can only come from someone who has been working on this directly, is that there is a limited production of the inactivated virus in cell culture system. So scientists have struggled in generating inactivated forms of this virus in general. So I think those are the three reasons: variability, glycosylation of the code proteins, and our limitation in terms of technique, that like how much virus we can generate, like deactivated virus.
Yes, and I would just like to add that presently there is no proper knowledge. Even if we have some candidates, it's difficult to screen different vaccine candidates, because only humans and certain monkey species, chimpanzees, can get infected by this virus. So
Yeah, so the lack of animal
lack
models.
of
I see.
animal models, yeah.
I see.
True. So I don't have really any more questions, but if you have any questions, we can discuss that, or in the audience, if we get any questions. Otherwise, we'll wrap it up. So we kind of discussed and Das also kind of discussed the various treatments, and yeah, we kind of covered different aspects. So I think, yeah, we got some question. We know that viruses and bacteria evolve over time with the exposure of antibodies to them. So is there a chance that in future the hepatitis C virus may not be affected by the vaccine, or the possibility of a new strain due to evolution of the virus? So first, I think vaccines are not yet in for hepatitis C. So yeah, that is one of the problem with this virus, that it's an RNA virus. It mutates readily, and it's difficult to develop a vaccine for that. Possibility of a new strain due to evolution of the virus. Shalini, do you want to answer that question?
So I'll just start with the first part. In case of normal drugs, the antivirals which are used against hepatitis C virus, resistance has been known. So virus does mutate because it has error-prone replication machinery. The RNA-dependent polymerase doesn't have proofreading activities, and in one day it can produce almost 10 to the power 12 variants. So that gives a lot of scope for variation. So antibiotic resistance is known. So mostly the treatments include a combination of different antivirals against different proteins of the virus. And evolution of new strain, I'm not sure. I'm sorry, I'm not very well aware of the biology of this virus, so I cannot say whether new strain can evolve or how easy it would be for a new strain to evolve. But it has many genotypes, as Dr. Sarkar already mentioned and Dr. Soumitra Das also mentioned, and each genotype has a different epidemiology, and different treatment combinations are required for different genotypes.
Yeah, absolutely. So if we don't have any more questions, I would like to thank Shalini and Shondipto for joining us. And again, thanks a lot for joining in. And thanks to Science Club of ISER Kolkata and Coglo 137 for organizing this wonderful event. Thank you.
Yeah, thank you all. Thank you all.
Thank you, everyone.
I request all the organizers and the speakers to stay after the event. We'll take a short snapshot. Can everyone else please switch on the cameras? We'll take a short screenshot. First of all, I would like thank all of you for attending this talk and hosting it. It was a pleasure to host all of you. It was a very interesting talk.
Yeah, true. I also enjoyed Dr. Das's talk, and thanks for inviting me.
And yeah, and thank you all for organizing this. Basically, we need more of this.
Yes,
We are planning to host more like these, more talks.
Yeah, we definitely need more of this in YouTube. And all I see in YouTube's are cooking videos, like which is important. But I think science videos should be
basic
also
science
there.
is also important. And people should be aware of these diseases, these facts. It's important that they know.
Yeah, yeah. And I think you should also like the young guns in ISER who are also a bright future, right? You guys should also be more visible, like now you are.
More involved. Ask more
Yeah,
questions.
actually, there were multiple exams scheduled today during
I
the
see.
talk,
Oh.
so many could not
Yeah.
attend. We called them.
No. I am giving advice to you just because I am on the other side now. If I had been a student with an exam coming ahead, I would have been like, who will go for a science club? This thing, yeah, I know. I mean, this happens.
And I understand.
But having said that, wonderful. Like this is something we need more.
Yeah, I really enjoyed it. Thank you all. Thank you.
Yes, thank you everybody for joining. Thank you, Ritama, for helping us out again.
No
And
problem.
thank you, Shondipta. Thank you, Shalini. Okay, we'll upload this video. The meeting was recorded. We'll upload it on YouTube and we'll share the links with you.
Okay,
Yeah.
perfect.
Thank
Thank you.
you.
Thank you very much.